So, as I say, welcome. Thank you all so much for joining us on this hopefully sunny where you are Friday evening. I'm Ella, I'm the Research Programme Manager at the TSA, and I'm delighted to be hosting this webinar this evening with the first of our CCORN grant awardees, Dr Elaine Dunlop, and I'm going to be co-hosting with a member of our Research Volunteer Network, Philippa Ward. So, this CCORN Award was one of four awards that was provided to researchers as part of our Research to Real Life campaign, where we're hoping to bring more research about within the UK and abroad to ensure the best progress within research for the TSC community. So Elaine was one of four awardees, and over the course of the next couple of months, we will continue with short webinars to introduce you to the various fantastic projects that were awarded. So before we start today, we would just like to say thank you so much to everybody who has engaged with the campaign, who has donated, who has signed up for the webinars, and we're really, really pleased to have you along on this journey with us. So I will hand over to Elaine just now, who's going to introduce us to her project, and then we'll open the floor up for questions afterwards.
Thank you, Ella. So yeah, as Ella said, I'm Elaine Dunlop. So I'm a senior lecturer at Cardiff University. I've been working on TSC since I moved to Cardiff, essentially. Initially sort of under Julian Sampson and Andrew T, who you may have heard about in TSC research and in the clinical side of things. And then, yeah, being able to sort of maintain the research in this area and kind of spin off into new areas that are kind of little bit more niche. So yeah, I'm delighted to be able to speak to you all tonight. Really excited about sharing our project with you. Feeling a little bit of pressure being the first up of the awardees, but I'll give my best go. So I've got a few slides that I was going to kind of think some people are quite visual. I like to see things, not just hear me chat. So I'll share a few slides just to kind of take you through a little bit of the background to the project, where we came from and what we're hoping to do with it. And then, as Ella said, open for lots and lots of questions afterwards. So I'll just see if I can get Zoom to behave and share my slides. So can people see that okay?
That's perfect.
Yeah.
That's come up, Elaine. Thank you.
Lovely. So yeah, as I say, this is a CCORN project that's going to be for 12 months. And the official title is EV RNA cargo as a potential TSC biomarker, which when I look at it is just lots of jargon. And so in simple terms, and probably the title I should have given the project is essentially diagnosing and monitoring TSC. Can we make it simpler? That's essentially what it boils down to with this project. So you'll be well aware of all the various challenges that there are in TSC, right from the diagnosis side of things to, monitoring and supporting the patients as they go through the journey, getting the appropriate treatments in place. But the challenges I'm particularly going to focus on tonight are around sort of diagnosis and monitoring of disease. So we already know, in that 10 to 15% of patients, when they have all the clinical signs of TSC, we can't always find that mutation in the TSC1 or TSC2 gene. So it'd be really nice to be able to have another kind of confirmatory way to say, yes, we're happy with this diagnosis. So can we come up with other ways to support the diagnosis for these patients? And then obviously the bigger challenge that affects everyone is thinking about monitoring of the tumour burden. From the NHS point of view, it's obviously expensive. It needs the specialist equipment. From obviously the patient and family points of view, it's the time, it's the hospital appointments, it's that extra anxiety with these different tests. So how can we go around making this monitoring and predicting of disease burden a bit easier for everyone? And so about 10 years ago now, there was kind of a lot of the experts in the TSC field from both the research side and from the clinical side who came up with kind of the future directions for where they thought TSC needed to go. And one of the areas they came up with was developing clinical biomarkers for TSC. And that's essentially what this project is aiming to do. So I'll talk you through a little bit about what a biomarker is and why we're interested in it. So essentially, a biomarker is just the name for any sort of biological molecule that we can detect in the blood or other body fluids, or indeed in your tissues. And it can be a sign of either a normal or an abnormal process. So in the past in TSC, there's various groups have looked at blood samples and tried to pick out differences and things that might be sort of representative of the TSC condition. Other groups have also looked at brain scans or brain waves. That's especially relevant when you're thinking about things like epilepsy in TSC. So those could also be potential biomarkers. And biomarkers can do different things. So essentially, in this project, we're looking for something that will allow us to detect differences in a TSC patient sample compared to someone who doesn't have TSC. And ideally, in the future, something that would help us monitor TSC over time. So essentially, at the moment, the types of biomarkers this project is going to come up with, or we're hoping it will come up with, are in the diagnostic field. So thinking about different things that could help support that initial diagnosis of TSC. But other biomarkers might be linked to the prognosis or the prediction of a disease. So how a disease is going to change over time, or how well it will respond to different therapies that might be given to a patient. And we can also have sort of more monitoring biomarkers that you can just measure over time and would be linked with, the status of the condition at any given time. So a little bit of background to where we came from to end up with this particular CCORN project. So back in 2018, we had an award from the TSA at that point to fund a PhD student who was called Mirren. And her project was to look at these interesting little things that are released by cells called extracellular vesicles. And that's where the EV part comes from. So people have known about extracellular vesicles for quite some time, but at the time we started this particular project, well, initially, actually, when we came up with the initial idea, we were struggling to find any papers on it, but one paper was published just before we started the project, looking at these vesicles in TSC. And I'll hopefully try and explain why they are of interest. So if this is your cell on the left-hand side, hopefully you can see my little mouse. So this is your nucleus with all your DNA. Our cells are obviously packed full of lots of different proteins. They've got lots of different receptors on the outside. And these vesicles are released by this parental cell. And they're kind of like bubbles, essentially, of material that get released into the environment around a cell. And they're going to be, their content is reflective of the content of the cell that they've come from. So they'll be full of proteins and RNAs, and I'll go into that in a little bit more detail. But the idea is hopefully there's some correlation between what's in the EVs and what was in the cell to start with. The idea being that obviously if you've got a cell which has alterations within it, whether that's a disease or a genetic condition, then any changes that are in the cell will hopefully be mirrored in some changes in these vesicles. And so these vesicles can either remain local to the cell that they've come from, and there's evidence to show that they can signal to other cells nearby and alter the behavior of nearby cells, which can be important when we're thinking about how tumours grow and how they interact with that tissue environment that they're in. But the other thing happens is they get distributed through the body. So they end up in our blood, they end up in our urine, they end up in other fluids. And all our cells are making these, so the normal cells and the disease cells. So we've got a real mix going on. And what we did with Mirren's PhD project was start to look at some of the protein changes that we see in these vesicles. So here on the left-hand panel, we've got proteins that were detected in our TSC2 expressing, so kind of our healthy cells, as much as cells can be healthy when we grow them in the lab on plastic. And on the right-hand side, we've got the proteins that came from our TSC2 deficient cells, i.e. mirroring what we see in some TSC cells. And essentially each of these pairs of black dots is a different protein. And we were looking for proteins that were linked to cell growth. So this is kind of a panel of different proteins we might expect to see altered in different growth conditions. And hopefully the take home from this essentially was on the right-hand side where I've got our cell that's mimicking TSC disease, the vesicles that are being released by that cell have more of these kind of proteins. You can see more of these dark black dots. And so Mirren's project built on this. We looked at some patient samples and we were able to show that some of the changes that you saw in the cells from the lab were also able to be detected in blood samples from the TSC patients. And so we were able to, a couple of years ago, publish some of this protein work. So why do we think EVs are useful for biomarkers? Obviously, I've said people have looked at TSC blood before to try and find biomarkers. Why do we think vesicles are going to give us a better chance of finding something useful? And the reason is because we can separate those vesicles out from the rest of the blood components. So our blood's full of all sorts of things. Apart from obviously the red blood cells, we've got immune cells, we've got lots of hormones, cytokines, fats and cholesterol, all sorts of things that can interfere with how we can detect changes. So the advantage with EVs, and why other groups and other diseases are also interested in looking at EVs as a biomarker, is that you can purify those out of the blood. You can kind of get rid of all that noise from the other components and hopefully hone in on some of those differences that have come from the cells originally. So if we zoom in on one of these vesicles, this is kind of the idea of what you've got. You've got various different proteins that are kind of embedded in the membrane of the vesicle, lots of other different types of proteins that are inside. And then of interest for this particular project are the RNAs. So the RNAs are the bit before you get to your protein. You might want to think back to your sort of biology at school. You've got your DNA, that makes your RNA, and that makes your protein. So we've done a bit of work, as I said, on the proteins. We've kind of progressed them a little bit further along the pipeline. We haven't done so much with the RNAs. And these RNAs can be a few different types. So I won't go into the details, but essentially they all have slightly different roles in the cell. But we could essentially detect any of those. And essentially an RNA biomarker could be just as good, if not better, than a protein biomarker, or even we might want to combine those in future to kind of give us more distinction. So the idea of this project is to look at these RNAs in the vesicles from healthy donors and from TSC patients. So we had a little bit of pilot data. So the TSC Alliance, which is kind of the TSA equivalent in the US, they've got a biobank of lots of patient samples, which is really helpful. And we were able to get, as you can see, just a small number of samples, extract the vesicles from the blood samples, and then look at the RNA through a process called next generation sequencing. The idea of that is it kind of looks at all the RNAs that we can possibly detect in the sample. And in this figure, anything in blue is not expressed very much, so it's low level. Anything in yellow is much more expressed. And hopefully you can see that there's a pattern. There's some RNAs here that we see more of in the TSC patients and some that we see less of. None of them are absolutely perfect. We're all human, we're all very variable, and there's definitely differences between us all. So the challenge is to try and pick out the ones that we think are gonna stick, are gonna stay the same. And that's really where this project comes in. So we've got that initial data, but we want to kind of push it forwards towards something that's going to be more clinically relevant and more likely to be a biomarker for the future. So the first challenge is to try and look at our data that we've got so far and pick the RNAs that look promising. So this is an example of one that we might pick out from our samples. It looks like it's lower in the control samples compared to the samples that have come from the TSC patients. And so we're trying to pick out a few of those that look like they've got promising differences to go from. The next stage is to try and A, look at a bigger group of samples. Does it hold true when we look at more patient samples? And B, see if we can detect these RNAs using a simpler technique. So the panel I just showed you was next generation sequencing. It's cheaper than it used to be, but it's still relatively expensive to do lots of samples on. And it takes quite a lot of time and effort to decode all the data that you get out of it. So if we can pick out our particular RNAs, we know exactly the ones we want to look at, we can use some simpler methodology to look at that. So the next challenge is really, can we still detect those changes using this different methodology? And then the final part of the project is kind of the really exciting bit that can hopefully help push us towards the clinic, is to work with this Welsh bioscience company called Melomics. So they have a platform that they're developing, which the idea eventually when they kind of get it all up and running is that you can go from a sample, so your blood sample to start with, right through to sequencing of it as an automated process. So be quicker, be easier. You don't need all the technical skills. Someone could be trained just to use the platform without needing all those technical sort of RNA skills in between. And so we're kind of working with them to see how their platform might be adapted to kind of look at vesicles, to look at RNA changes, and whether that might help streamline any findings that we get in the research lab into more of a clinical test. So yeah, that's essentially what we're hoping to do over the next few months. So happy to answer any questions or explain anything that I've kind of skimmed over. And yeah, love to hear your thoughts on it.
That's lovely. Thank you very much, Elaine, for sharing the overview of what the next 12 months are going to look like within your lab. I have one question before I maybe open up to the floor. I have a couple, but I'll start off with one if that's okay. Um, what would success look like at the end of the 12 months for this project? And we appreciate that, of course, within lab work and within this space, sometimes we don't find what we expect. And that is still incredibly useful, isn't it? But standing now at the beginning, at the start line, where would you hope to be in 12 months?
Yeah. So I would hope that we've been able to pick out, I don't know, 4 to 6 RNAs that are kind of holding up to a bit more scrutiny when we look at more samples. I would be hoping that, well, we would see whether we're able to detect them sensitively enough with the different technique, because obviously if we change techniques so we just can't detect them anymore, that's not going to be very helpful. And getting a better sense of whether any of them are going to be like standout biomarkers, i.e. one thing that we just need to test for that one molecule and it's going to be, the answer, or actually with whether we need to combine them in some way to get something more meaningful, and thinking about how we might do that combination scoring. And I guess the other thing is really to maintain this collaboration with Melomics. So this is our first time working with them. So this is really exciting to be able to, bring this project to them. So hopefully to kind of maintain that collaboration with them, see how their platform develops, see how this would fit in with it, and just kind of keep that open dialogue to see, to really push forward translation eventually to the clinic if we can.
Thank you, thank you. I'll open up the floor just now. Does anybody else have any questions they would like to ask about the project?
I would, if that's okay.
Yeah, go ahead.
Apologies if I missed it, but at the beginning you mentioned the kind of mosaic TSC. Is this kind of test that you're working towards going to be applicable across kind of all three formats, I guess, if for want of a better word, of TS then? So the diagnostics for 1, 2, and then the kind of currently unable to be diagnosed.
Yeah,
patterns across all of them.
That's a really good question. So ideally, yes, we would love something that is just going to be a kind of a catch-all, and it doesn't matter what your mutation is, or even if we can't find the mutation, we're still able to pick up these kind of biochemical differences. The reality for this project is we will probably start just with samples that have come from patients with a TSC2 mutation. That's just to kind of streamline things and kind of, generally, obviously, more patients tend to have.
Yeah, it's your bigger population, really, isn't it?
It gives us more samples to work with to start with. So that'll be our starting point, and that's what we did with the protein work that I showed a little bit of. What I didn't show you with the protein work today, just in the interest of time, but that we're hoping with Jessie, who's the postdoc on it with me, we need to write it up and get it published. But essentially, we have gone back with our protein work to also look at patient samples from TSC1 mutations that have, patient samples where the patient has a TSC1 mutation, and have been able to show actually our protein differences are holding true no matter which, whether it was a TSC1 or a TSC2 mutation sample, which was lovely to see. So yeah, I'd say with this one, we'll start with the TSC2 just because it gives us that bit more scope and a few more samples to work with. But yeah, we would definitely want to then go back and recheck other samples, and indeed those NMI patients, because actually if this is only matching up with a no mutation already, then it's not adding to that field of information for those NMI patients. So yeah, it's really important to go back and check these things. So yeah, it's a really good question.
Brilliant. I've got a sort of further question, just building on Ella's question, where she asked you where you'd like to be in a year. In an ideal world, where does this end up in the future, and how much shorter could diagnostic time take? Because at the moment, the DNA and the blood screening and everything, it's they explained it to me like it's looking for a typo in an encyclopedia, so it can take an unknown amount of time, which when you are living with that hanging over you as a family waiting for a diagnosis is awful. And if it's going to mean that you haven't got that but you've got something else, then I don't know if that adds on even more time. So what's the sort of utopia for this?
The utopia. So obviously the DNA technology has moved on so far, so they're able to give a quicker genetic diagnosis now anyway than in the past. But yeah, ideally this would be a test. I mean, if we could get it to the stage where we isolate the vesicles and we isolate the RNA from those, and then we run the qPCR. I mean, we could certainly do that within a week, probably less than a week, to be honest, if we had it streamlined. And yeah, I guess the other challenge, I didn't mention it, but the other part of the project at the very end we're hoping to move towards is obviously the isolating the vesicles is an extra stage, an extra time and effort to do that. And while it's great to help us kind of, as I say, remove that noise from the samples and hopefully hone in on what the differences are, actually, once we detect some differences, we might not need to purify the vesicles anymore. If we know what we're looking for, we might be able to just look at a whole blood sample and go, yes, it's high or it's low. So actually part of the end of this project, assuming we're able to pick out some differences from the vesicles, is to actually just go back to a plasma sample from a patient and say, can we still see those differences if we don't bother with the purification step? Because if we could get rid of that, that simplifies it even further as well, and obviously speeds up the process too.
Well, thank you.
Thanks, Elaine. Carol.
Hi. So I'm trying to keep up with the talk.
Yeah. Go ahead.
You talk quite quickly.
I know, it's a weakness.
So if I'm getting it right, obviously when my daughter was diagnosed with TS 30 years ago, she was 5 because things were not advanced then, and it was a long time before GPs joined the dots. So she had a blood test after it was, already kind of decided she probably had tuberculosis because she had the CT scan and it showed the tumours. And so do you think there will be in future? I know that TSC is rare, but its effects can be devastating, obviously. And we know, we've lost 2 lovely young adults already since Christmas. Is there a time, do you think, when diagnostic screening testing will happen when pregnant mothers are being screened for other conditions? Do you think because you mentioned mosaic, and obviously a lot of people don't know they have tuberculosis until they have their first baby.
Yeah.
Because they didn't think to get screening or whatever. Once people have had that baby, then things have moved on a lot, haven't they? And you can get screening. But do you think we will enter a world where mothers are screened for various diseases, a whole spectrum of diseases while they are pregnant, before the baby is born, and that would include maybe TSC?
Yeah, it's a good question. Yeah, how early can we diagnose these things, and can we give people answers earlier? So I would say, obviously, the challenges at the moment would be how much of that fetal material would end up in the mother's bloodstream to be able to detect changes in the first place. I know there is some, there is evidence of fetal cell-free DNA and things ending up in the mum's bloodstream. That's sort of the NIPT test, which is kind of the new genetic test that they use for screening pregnant women for some of the trisomy conditions. Essentially relies on that fact that you've got some fetal material in the mother's blood. It's going to be a case of detection. Obviously, they've been able to pick out an extra, chromosome, but that's a big chunk of DNA that's altered. So yeah, narrowing it down to a gene difference and knowing which particular gene you want to look at is going to be trickier. I'd say in the more immediate future, earlier diagnosis is possible. I don't know whether you've heard of the Generation study, which is a Genomics England study, for anyone who's not so familiar with it. So obviously at the moment, babies get their heel prick test, about 5 days old or so, and they test for a few conditions within that. So there's an ongoing study with Genomics England called the Generation study, where they are taking cord blood when the baby's born and screening that for, I think it's just under 300 conditions now, where they know early diagnosis is crucial to make to improve the life of that baby, where there's some sort of management or treatment or something like that in place so they can take action for that baby. So that's going through at the moment, and they're picking up a few babies earlier from that. I saw actually there was a genomics conference yesterday in Cardiff, and they were saying that, I can't remember the exact numbers, but 40 odd babies have been picked up through that, who would also have been picked up through the heel prick test, so the kind of those conditions, but they've picked up another, for want of a better number, around about 40, it was sort of similar-ish numbers, where they wouldn't have been diagnosed because that wasn't screened for in the heel prick test. So that's 40 babies who've been diagnosed with some of these rare conditions a lot earlier than they would otherwise have been. So I think something like that could potentially come forwards. I guess the question with all of these things is kind of that ethical. Do you want to know early? What's important to know early? Should we be screening for all these things? And there's a lot of ethical issues, and obviously everyone has a different viewpoint on what might be helpful or not helpful for them. So yeah, I think there's a lot of work to be done in that space as well as to what people would find acceptable and what society would find acceptable.
Thanks, Elaine. Thank you. I'm conscious of the time. We have a couple of minutes left. Does anybody have any final questions for Elaine before we close off this evening? Pooja.
Yeah, I was just going to say, it's a great question, Carol, but the problem is that I attend a lot of policy meetings and advocacy meetings, and the issue is not so much as about the conditions being screened, either, in vitro or a newborn screening. I think the problem is, the main problem is around ethical concerns of these things, and that really is something that none of the political parties are, willing to risk. So informed consent, autonomy, they talk about best interest of the child. And I think their main concern is about false positives, overdiagnosis, and anxiety. So those are the kind of, uh, things that, again, have hampered the effort to open up, the screening to other conditions as well. I mean, we know that TSA was not included in the newborn genomic program because of the variability. So they are initially looking at conditions that have a certain, trajectory, like a defined pathway. And they felt, or the committee felt, that including TSC, probably, I mean, we argued against this, but they said they won't benefit as many newly diagnosed children because up until the age of 2 or 5, you just don't know which direction the TSC is going to go. So it's going to raise more anxiety for the parents, whether their child will develop infant. So they will always be looking for symptoms. So yeah, these things are just, they get left out on ethical and other issues.
Yeah, I mean, TS doesn't have a prognosis, does it?
That's the thing. Yeah, exactly.
I mean, I remember Anne Carter saying all those years ago, in effect, parents live in fear. But I would say on the ethics side, my son is 38. And when our daughter was diagnosed at 5, her father and myself were screened and apparently not TS. And they wouldn't screen our son because they said he wasn't 18. He had to give his own permission.
Exactly.
Well, my son is now 38. He hasn't had any children yet. I don't have any grandchildren. I just have a grand cat. And I keep saying to him, get the blood test. And I would have to say to you, Pooja, that the thought that my son might marry and have a child with tuberous sclerosis is, for me, I would be devastated. I just feel our family could not accommodate another baby with tuberous sclerosis. I think its effect, on a family is
No, absolutely. And I'm sure that
huge.
because of a diagnosis in the family, he will be eligible for genetic counselling. So
He is, but he's just a pain. He doesn't get round to because he's a bloke and won't turn up at Nottingham Hospital to get his blood test. And I've given up.
We're in a similar boat, Carol, because my son with TS, it turned out that I found out at 35 I've got it a lot of years ago now, but my other son, they won't let him. They've screened everybody else in my family, my brother, my cousins, all sorts of people, but the one person who's got a 50% chance of having it cannot be screened until he's can consent at 18.
I know,
mad.
and
it's absolutely bonkers as far as I can tell. That's an entirely different conversation, isn't it? But
because if they had TS, they could still develop some, and I don't know medically what the word would be for it, but some
complications, maybe.
Complications, and usually it's kidneys. or in women it could be lungs. And if they don't know if they have TS, it's quite dangerous, isn't it? You know, like you're not supposed to have an X-ray if you have lamb in the lungs. So, you know, people need to know. And my son is an example of someone who's just meandering through life and has got to the age of 38 with his sister, who we know has TSC2, and we don't know if he has TS or not. And it's maddening. But I really feel, and I hope that eventually women can be screened by a simple blood test, you know, before they are 12 weeks pregnant, that would show a range of diseases, especially in something where there are genetic changes. It should be scientifically possible, I would have thought, in the 21st century.
I
I
think it
think that's what we
is,
should be aiming for.
to a certain extent, it is possible, but I've also heard arguments, especially in the US, because of the private medical system. The implications for medical insurance. If the insurance company gets a whiff of any, predisposed or, what's the word, condition, that people will be denied, their families would be denied. So I think, but for UK, I can assure you, the Specialized Healthcare Alliance, they're pushing for more conditions to be included, and TSA. The deadline is 1st of May, and together with Francis Emsley, I am drafting a response on behalf of the TSC and TSC community to see how we can push for, at least the TSC to be included in newborn screening.
Thank you for bringing that, Carol, and sharing your experience, and also sharing your experience, Philippa. And I think it just does highlight the importance of this project to bring diagnosis about sooner, starting where we are now with the things that we can, so start to work on with the science, with the ethical things aside, as a temporary, so that we can continue to make progress forward. I'm going to thank Elaine very, very much, for sharing your project with us. We're really, really excited to see how it develops and evolves over the next 12 months, and we look forward to sharing lots of updates with the community. Thank you all very much for joining this evening, sharing your Friday evening with us, and we hope to welcome you at our next webinar, which will be in May, date to be confirmed, and we will send emails out to the mailing list shortly. Thank you all very, very much. And thank you,
you.
Philippa, for joining us, and translating that, the science and the community boundaries. So thank you. Special thank you to Philippa. Have a lovely weekend, everyone. Hope you enjoy the
Thank
sunshine
you.
where you are.
Bye.
See you later. Bye now.
Bye.